Hello everyone, I welcome you all to the 5th lecture of this course. So today we are going to see characterization of nanomaterials. So there are several methods available for characterization of nanomaterials, but in this lecture we are going to focus on UV visible spectroscopy and dynamic light scattering DLS and zeta potential, and we are also going to focus on electron microscopy like TEM and SEM and atomic force microscopy. So when you synthesize any metal nanoparticle, the first thing you observe is formation of color. So what is the reason for the beautiful colors? The reason is surface plasmon resonance. So in the previous lectures I already explained what is surface plasmon resonance. Let us see once again. So usually the bulk metal is shiny due to the electron cloud. So these electrons have dual nature. So if the light of matching wavelength produces resonance, then electron clouds start vibrating. So for nanoparticles, this wavelength lies in the visible region. So that is why you are getting these kind of colors. So when the incoming radiation induces the oscillation of conduction electrons, which leads to surface plasmon resonance. And when you study these metal nanoparticles under the UV visible spectroscopy, you will get different kind of peaks, depends on the size as well as shape of the nanoparticle, and there will be rep shift with increasing in the particle size. So when you study the metal nanoparticle in UV visible spectroscopy, for example you can take the example of gold nanoparticles. So usually the gold nanoparticle is ruby red color, and if the gold nanoparticle is agglomerated or aggregated, it will be like a purple color, and if it is studied under the UV visible spectroscopy, usually the dispersed gold nanoparticles will get peak at around 550 nanometer, and the agglomerated or aggregated particle it will give peak at around 650 nanometer. So any metal nanoparticle, the first step of characterization is your UV visible spectroscopy, and for any nanoparticle the stability and size distribution is very important. Okay, the stability of the nanoparticle can be studied by using zeta potential measurement. So what is zeta potential? The zeta potential of the particle is the overall charge that the particle acquires in a particular medium, that is called as zeta potential, and the zeta potential value should be plus or minus 30 millivolt, and or it should be above the plus or minus 30. So that indicates the good stability of the nanoparticle, and the zeta potential depends on the pH and electrolyte concentration of the particular material, and this is a zeta sizer equipment. So and for zeta potential we have to use this kind of cuvette, and when you put this cuvette inside this zeta sizer, it will give the zeta potential of your nanoparticles. So let us see dynamic light scattering. Using this same equipment we can also measure the dynamic light scattering of the particles. So this is for direct determination of particle size in solution. If you want to measure the particle size in solution, we have to use the DLS, that is called as dynamic light scattering. So here the measurement is based on the principle of DLS, and here the size of the nanoparticle will include even the stabilizers bound to the molecule. Okay, so which cannot be seen by the TEM, and here the principle is the scattering of laser light is due to the Brownian motion of the particles. So due to which what happens is there will be temporal fluctuation in intensity, so which measure the hydrodynamic diameter of your particle, and it is very sensitive to other biological molecules, and it can measure 0.6 nanometer to 6 micron. But the drawback of this DLS is we cannot get any information about the particle shape. Okay, so you can see here this picture in transmission electron microscope TEM you will get the particle size, exact particle size, but in case of DLS, that is dynamic light scattering, you will get the hydrodynamic diameter. Okay, so the particle size will be slightly bigger because you are determining the particle size in the solution. So before we understand what is zeta potential, let us see some of the definitions. So this is your highly negative colloid metal particle. Okay, so this is your particle surface, the blue color is the particle surface, and the red color is the stern layer. This stern layer is a rigid layer of ions which is tightly bound to the particles, and these ions travel with the particle, and this black colored minor line is also called as slipping plane, or it is a plane of hydrodynamic shear. This is the boundary of the stern layer, and ions beyond this shear plane do not travel with the particle, and this green color is the diffuse layer, also called as electrical double layer. So here the ionic concentration not the same as in the bulk. So there is a gradient in concentration of ions outward from the particle until it matches the bulk. So, let us see what is zeta potential. The electrical potential that exists at the slipping plane is called as zeta potential, and this zeta potential mainly depends on the stabilizing agent as well as the coating agent or capping agent. Okay. So, the magnitude of the zeta potential gives an indication of the potential stability of the collider system. Suppose if all the particles have large zeta potential, that means if it is more than plus 30 or if it is more than minus 30, that is it will repel each other. So, the particles having uniform charges will repel each other and there is a dispersion stability. But suppose if the particles have low zeta potential values, that is there is no force to prevent the particles coming together. So, the particles come together and it will aggregate or agglomerate. Okay. So, that is why they made a dividing line between stable and unstable aqueous dispersion. So, which is plus 30 or minus 30 millivolt. So, if you have your zeta potential plus 30 or minus 30 or above, that means your particle is highly stable nanoparticles. And again this zeta potential is highly dependent on the pH. So, in this example you can see here. So, as I told you earlier, so the plus 30 or minus 30 above is the stability of nanoparticle will be very high, very good. So, in this example you can see here, so the pH below 4 as well as pH above 7.5, the nanoparticle is stable in this example. So, let us see how we can use microscopy as a means of nano characterization. So, first what is microscopy? So, microscopy is any technique for producing visible images of structures or details which is small or otherwise we cannot see by the human eye. Okay. So, why we need microscope? Because in order to effectively study something or build something, we need microscope. Okay. So, we can understand the structure, shape and everything. Further we can understanding the structure, we can modify the structure or shapes. Okay. So, as the things we are interested is getting smaller and smaller, we need a more and powerful microscope to understand the nano structures. So, what are the things you will understand by this nano characterization? So, you will get an idea about what is the dimension and what is the structure of the nanoparticles, and also you will get an idea of what kind of material it is made up of, what are the elemental proportions and what are the physical chemical properties. So, these are the things you can understand by nano characterization. So, let us briefly see the origin of microscopy. So, in the first century Romans invented the glass, okay, and they are experimenting with various shapes and converging lenses. In 1590 the Dutch eyeglass maker, so they made a compound microscope, and in the mid of the 17th century Anton van Leeuwenhoek, he first observed the bacteria and protozoa under the small microscope, simple microscope. And in the 17th century Robert Hooke, so he made a lot of modification to the microscope and they made a kind of compound microscope. Okay. So, the next 200 years this optical microscope, it played a very major role in the biology and other fields. Okay. So, in the late 19th century Ernst showed that improvement of the magnification of the optical microscopes was fundamentally limited by the wavelength of the light. So, the wavelength of the light is very important in deciding your magnification of your optical microscopes. So, that is why they moved to electron microscope. Okay. So, in 1931 Ernest Ruscott they invented the electron microscope, and in 1938 using the TEM electron microscope they reached a resolution of 10 nanometer, and 1940 2.4 nanometer resolution, in 45 they reached 1 nanometer resolution, and 1981 so these people have invented scanning tunneling microscope, and in 1986 atomic force microscope was developed by the same group. So, let us see the basic principle between the SEM and TEM. So, here the SEM scanning electron microscope, it is similar to your scanner. So, it will scan the sample and it will give the image, and in this TEM the transmitted electron microscope, so the electrons will be transmitted and you will get the image. So, this is the picture of the transmission electron microscope. So, there are two modes, bright field as well as dark field. So, you will get this kind of images using this transmission electron microscope, and so here the electron microscope, the electron pass through a very thin sample to form a image. In a simple term we can understand it is similar to the your slide projector. So, in TEM, that is a transmission electron microscope, it is a complex viewing system equipped with a set of electromagnetic lenses, okay, and which you use to control the imaging electrons in order to generate the extremely fine structural details that are usually recorded on a photographic film. So, here the eliminating electrons pass through the specimens, that is why the information is said to be transmitted image, that is why we are calling it like transmission electron microscope image. And the modern TEM can achieve magnification of even 1 million times with the resolution of 0.1 nanometer. And for transmission electron microscope, you need a specimen, very thin specimen, so that the beam can pass through it and it can give the information about the size of the nanoparticle, morphology, and also it can give the information about the crystallographic nature as well as compositional information. So, this is your TEM grid, very small size TEM grid, and the zoomed image of the TEM grid is this is the TEM grid. So, the TEM grid has two sides, one is shining side and non shining side. So, you have to deposit your sample on the non shining side, which is coated with the carbon. Okay. So, let us see the principle of this TEM. So, the principle is similar to your optical microscope, but here instead of using the light, we will be using this electron as the light source. Okay. So, the condenser aperture stops the high angle electron, that is the first step in improving your contrast, and this objective aperture and the selected area aperture are optional, but it can enhance the contrast by blocking the high angle diffracted electrons. So, what are the advantage of this? We can look at the non conducting sample, that is you can measure, you can study the size, everything of the polymer sample, ceramics as well as biological samples. And these are the basic systems of the TEM. So, the transmission electron microscope is made up of a number of different systems, okay, and it forms one functional unit of capable of orienting and imaging extremely thin specimens. So, it has the eliminating system as well as specimen manipulating system. So, in the elimination system consists of electron gun and condenser lengths. So, that give rise to and control the amount of radiation striking the specimen, and in the specimen manipulation system it is composed of specimen stage and specimen holders and also related hardware which is necessary for orienting the specimen inside or outside the microscope. So, here you can see here this is a variable aperture holder of TEM. So, your TEM grids will be placed here and you can keep this O-ring seal, so that will permit the aperture to be sealed off inside the vacuum of the microscope column. So, here is the example of your TEM image, how it look like. So, when you study this cellular nanoparticle under this transmission electron microscope, you can see here this black color dots are your cellular nanoparticle, and as I told you in the previous lecture, we have to include the scale bar. So, using this image J software, we can measure the particle size. So, in this case the particle size is approximately 8.2 nanometer in diameter. Also, we can study the diffraction of your sample by switching your sample from image mode to diffraction mode, and which can be easily achieved by changing the strength of the intermediate lengths. So, when you see the electron diffraction pattern of your sample, so this diffraction patterns give crystallographic information about your material, okay, and it can determine whether your material is amorphous or crystalline or polycrystalline, and it can measure your crystallographic information quickly as well as effectively. In case of amorphous material, you will get this kind of diffused ring, and in case of crystalline material you will get this kind of spotted, and in case of polycrystalline material you will get the rings with this kind of bright spots. So, how we are getting this kind of spots in the polycrystalline material? You can see here, in case of monocrystalline or crystalline material you will get this kind of spots, and when you combine this kind of things you will get rings with the bright spot, that is called as polycrystalline material. Another example of diffraction pattern you can see here. So, in amorphous carbon you will get the diffused ring pattern, and polycrystalline aluminum you will get this kind of rings with the spots, and single crystal gold you will get this kind of bright spots. So, the applications of TEM, so it is heavily used in material science as well as biological science. So, we can get the microstructural analysis and also we can study the crystal structure, and the magnification is very high here. So, that is the very advantage, but the another drawback is we need a very thin sample, and the sample should be transparent, and the thickness of the sample should be in the range of 200 nanometer. And let us move on to the other microscope, that is scanning electron microscope. Okay. So, this is a picture of scanning electron microscope. So, as I told you earlier, so here instead of transmitting the electron, here the sample will be scanned and it will produce the image. So, it is a microscope that produces an image by using electron beam that scans the surface of the specimen inside a vacuum chamber. Okay. So, here the same functions like an optical microscope, but as I told you earlier, it uses electron instead of light waves. Okay. and here the SEM uses a series of electromagnetic coils as lengths to focus and manipulate the electron beam, and here the sample should be in a completely dehydrated form and it should be made conductive. Okay. So, what are things we can study using this SEM? We can understand the topography and morphology of the sample, and we can also understand the elemental composition, and we can understand the crystallography and orientation of the grains. So, how the SEM works? So, here the electron beams follow a vertical path through the column of microscope, okay, and it makes its way through the electromagnetic lengths and which focus and direct beam towards the sample. Once the electrons hit the sample, backscatter electrons and secondary electrons are ejected from this sample. So, the detector will collect the secondary electrons or backscatter electrons and convert them into a signal that is sent to a viewing screen, and it will produce the image, and this is similar to your ordinary television, how it produces the image. And here the voltage should be like 0.1 kV to 40 kV, we can go in this range, and for biological sample it is 10 kV is recommended, and for a non-biological sample we can go even 20 kV. How the same works? You can see that there is electron gun passing the electron beam. So, it is hitting the sample and the backscatter electrons will be amplified and it will display the image. So, this is your SEM step where you have to prepare your sample. So, your sample should be dried, okay, and it should be placed on the top of this SEM step which is having this carbon tape. So, how do we get an image? So, when the electron hit your sample, so the backscatter electrons will be detected by the detector. So, it will scan your sample one by one, and that will be detected by the detector, all the backscatter electrons will be detected by the detector, and it will take it to the computer, and using the software it will give the 3-dimensional image. So, we can compare with the optical microscope versus scanning electron microscope. So, in optical microscope you will get the 2-dimensional image, for the same compound you are getting this kind of image, and when you study under the scanning electron microscope you will get the 3-dimensional structure. So, what are the advantages of using this scanning electron microscope? We can have a improved resolution, and also you can have a good depth of field, and the limitations is it required a vacuum, and also it is the equipment cost and otherwise expensive. So, another advantage is we can do the energy dispersive analysis of X-rays. The principle is each element has unique electronic structure and thus interacts uniquely with the electromagnetic radiation. So, it can be useful for chemical characterization of a substance. So, if you see this is your EDS results, that is a elemental analysis result. So, you will get the what are the elements present in your sample that can be measured using this EDS analysis. So, next we move on to this scanning probe microscopes. So, under this scanning probe microscope there are two types, one is scanning tunneling microscope and other one is atomic force microscope. So, here we can monitor the interaction between the probe and the sample surface. So, briefly on history of STM, the scanning tunneling microscope was invented by G. Binning, okay, and they awarded Nobel Prize in 1984, and after a few years they have invented this atomic force microscope, okay, and currently the AFM is the most common form of your scanning probe microscopy. So, in scanning tunneling microscope STM, what we are going to measure? So, we are going to monitor the electron tunneling current between a probe and the sample surface, okay. So, what is electron tunneling? It occurs over a very short distance, and the tunneling current depends on the distance between your STM probe and the sample surface. So, we are going to measure the tunneling current between your tip as well as surface of the sample. So, here the basic principle is tunneling. So, the tunneling current flows between the tip and the sample when separated by a less than 100 nanometer, okay, and this tunneling current gives us atomic information about the surface when the tip scans the sample. So, this is how the STM image look like. So, but the challenges with the STM is like it works primarily with the conductive materials, and there is a chances of vibrational interference as well as the contamination by physical like dust particles and other pollutants in the air, and also chemical reactivity of the samples. So, next one is atomic force microscopy. So, these three people invented this atomic force microscope in 1985, okay. So, using this atomic force microscope we can measure almost, you can image any kind of surface including polymers, ceramics, glass and biological samples, anything can be studied using this atomic force microscope, okay. So, this atomic force microscope monitors the force of attraction and repulsion between a probe and the sample surface. So, here the tip is attached to a cantilever, okay, which moves up and down in response to forces of attraction or repulsion with the sample surface, and this movement of this cantilever is detected by the laser as well as the photo detector. So, here you can see here this is your cantilever and this is your tip, okay, and if it moves on the surface up and down, so depends on that the laser beam position will be changed. So, those will be measured using the photo detector and it will give the image according to that, okay, and we can select the tip according to our sample as well as applications. So, the first one is normal tip and this is a super tip and this is the one is ultra lever tip, and here we can measure the force. This cantilever is designed with a very low spring constant, that means it is easy to bend, okay. So, it is very sensitive to even a small force. When the laser is focused to reflect off the cantilever and onto the sensor, so it will produce the image. So, here the position of the beam in the sensor measures the direction of the cantilever and in turn it force between the tip and the sample. So, how the force are measured using the Hooke's law, and here you can see here this cantilever tip, it is going up and down in the sample, and you can see here this the laser beam is also getting deflected according to the sample's nature, and if there is no interaction between the tip and the sample, the cantilever is not deflected. So, in this case you will get the laser beam will be detected in this area, and if the cantilever is deflected you will get the laser beam portion will be here, and if it is having repulsive force and the this laser beam portion will be here. So, based on that it will produce the image, and this AFM has 3 different modes. One is contact mode, non-contact mode and tapping mode. So, in this contact mode you will get a high resolution image, but the problem is it will damage to the sample because the cantilever tip is touching your sample, okay, and it can also measure the frictional forces, and in the non-contact mode it is a lower resolution, but it will not cause any damage to sample because it is not touching this sample, and in the tapping mode it will give a better resolution and it is minimal damage to the sample. So, you can see here in the contact mode the distance between the tip and surface is 0.5 nanometer, and in the tapping mode the distance is approximately like 2 nanometer, and the non-contact mode it can reach even 10 nanometer the tip surface separation, okay. So, in case of contact mode this cantilever tip is touching your samples. In case of tapping mode the repulsive force will be high, and in case of non-contact mode the attractive force will be low. So, let us see these modes in detail. In contact mode the tip will be in continuous contact with your sample. This is your tip and this is your sample, it is touching your sample and it can give the details of your sample, okay. So, it is mainly useful for hard samples, okay, and imaging can be done in air and liquid and it will give the high resolution, but the drawback is it can damage your sample or it can damage the cantilever tip. Next one is force spectroscopy mode. Here the consecutive cycles of tip approach and retract, and here we can measure the interaction forces, okay. And next mode is intermediate or tapping mode. Here your cantilever tip will be oscillating, it will be oscillating in a uniform frequency, okay. So, the tip touching the surface gently and frequently, and it is mainly useful for the biological samples, and here we can do the imaging in air and liquid and it will give a good resolution. And third one is non-contact mode. Here also will be having oscillating cantilever, but here the tip is not in contact with the sample, okay. So, it will be mainly useful for the soft samples. For non-contact mode you have to do the imaging in the vacuum condition, okay, and here the distance range will be like 50 angstrom to 150 angstrom. And how do you prepare the sample for the AFM? So, you need a completely flat and rigid surface. So, we can use the mica which is anatomically flat surface, and also you can use that SiO2 glass, okay, and the typical sample size should be like it can be like 1 centimeter square, or for liquid sample it should be between 1 microliter to 100 microliter. So, these are the AFM images of control cells and treated cells, okay. So, in the control cells you can see here the surface is very smooth, and these are treated cells. So, these are the mammalian cells, and treated with some kind of nanoparticles. So, you can see here due to the nanoparticle treatment the cell membrane get damaged. So, you can see here the roughness of the cell is getting increased. So, we can measure the surface roughness using this atomic force microscope, and these are the some of the AFM images. You can get an idea how the AFM images look like. So, let us see the applications of AFM. So, we can study the topographical surface, and we can also determine the roughness of the surface sample, and also we can measure the thickness of your crystal growth layer. And here the another advantage is we can image non-conducting samples such as DNA and protein. For most of the biological samples we will be using AFM, because for if you are using the SEM or any other microscope, so you have to do some kind of treatment, like you have to sputter with the gold, then only you can make your biological material to a conducting material, then only you can use the SEM microscope for imaging. But in this case of AFM, so you can even use the non-conducting surface protein and DNA, and we can also study the dynamic behavior of living and fixed cells. In case of electron microscope like scanning electron microscope or transmission electron microscope, we cannot study the living cells. Okay, so we have to fix the cells and it should be in a dried form, then only we can study. But in case of AFM, we can even also study the living cells, and it can be done in liquid or gas mediums, and here most of the mode vacuum is not required. Okay, and here we do not have to do any special treatment of the sample. As I told you, like for SEM you have to do the gold sputtering so that the sample will be made conductive, and here the resolution is also very high. So, we can reach 0.1 nanometer vertically and 1 nanometer in the x and y direction. As a summary, so in this lecture we have learned how to characterize the nanomaterials using UV visible spectroscopy, and also how to study the hydrodynamic size of the nanoparticle using DLS, that is dynamic light scattering, and how to study the stability of the nanoparticle using zeta potential, and we have also learned how to prepare sample for ultra microscope and how to study the sample using transmission ultra microscope and scanning ultra microscope, and we have also learned how to prepare sample for atomic force microscopy and what are the modes, various modes available in atomic force microscopy. So, I will end my lecture here. I thank you all for listening this lecture. I will see you in another lecture.